So chapter 7 is called How Cells Harvest Energy, but again, it's going to this idea of cellular respiration. So the first thing is this idea of reduction and oxidation reactions. The top two areas, is that reducing or oxidizing? And again, remember we showed you all earlier this whole little phrase called Leo goes Ger. Okay, where if it's losing electrons, it's oxidation. If it's gaining electrons, it's reduction. So what is it? What do you think? Oxidation? Good job. It is oxidation. And so because it is losing electrons, and you're like, well, what is the electrons being lost? Well, look, it's going from this C6H12O6 to the CO2. Okay, this carbon is now going over here. It's losing a hydrogen. And a lot of times, when you look at oxidation, it is truly dealing with the electron, but it's also pulling a proton with it as well, which is pulling and basically a proton is also a hydrogen, okay? Because hydrogen is basically just a proton. Okay, so it's pulling that hydrogen along with it. Since it's losing its hydrogen, it also means it's losing its electrons. So this is an oxidation. So what would this be? Reduction. Good. Down there on the bottom, that's an image from your book. It is showing how oxidation and reduction reactions do occur. Basically, what it's talking about is the NAD+, which is a part of the electron transport chain, which we're going to look at today. It does pull in, when it goes and connects to this enzyme, it grabs these extra hydrogens. These hydrogens are being lost, so this is actually an oxidation reaction. But then when the NAD comes back, it then becomes a reduction reaction. Okay? So in chemistry, a lot of times when we do redox reactions, we're looking at electrons. In biology, most of the time when we look at oxidation and reduction, we're primarily focusing on the hydrogens, not so much on the electrons. Okay? And again, the reason why is because electrons are pulling the hydrogens with it. So in oxidation, they lose technically hydrogens, and then in reduction, they gain hydrogens. So this oxygen is being reduced to water. Okay? This carbon dioxide is being reduced. This carbon dioxide was being reduced from that. So it's oxidizing, and these are being reduced. And again, the reason why, if this arrow was going the other way, this arrow was going that way, back towards it, this is respiration. What's this reaction that way? Photosynthesis. So this is respiration. So respiration, we're oxidizing glucose and we're reducing oxygen. Whereas in photosynthesis, we're oxidizing water and we're reducing carbon dioxide. Okay, so cellular respiration is just a process of basically stair-stepping electrons. Okay, so this image on the right is an image from your book. Basically, the process of respiration is basically taking phosphates, breaking them down, and then reducing them back to ATP, and just going through that cycle. Okay, so when you talk about ATP regeneration, if you had that as a project, Chapter 7 is also another good place to look. Both Chapter 6 and Chapter 7 is a good place for resources. This is what I was kind of showing you last time. This catabolism, the breaking down of phosphates, will form ADP and inorganic phosphate. The anabolism, where we phosphorylate ATP, or ADP, then forms ATP. So the entire process, and you do need to know this formula right here, this idea of glucose plus 2 ATP plus O2 will yield 30 ATP plus carbon dioxide plus water. So you do need to know that formula. Okay? And sometimes in these formulas, we'll pull the ATP out because technically energy is not matter. ATP is, but the energy from the ATP is not matter. It's just energy. So most of the time you'll just see glucose plus oxygen yields carbon dioxide plus water. Okay? You think back to your chemistry, this is actually a combustion reaction, right? Because if you take a carbon base plus oxygen, it'll always yield carbon dioxide and water. So basically, we're just a gigantic furnace. Here's the theoretical steps of ATP, the theoretical yield of ATP. So the theoretical steps of respiration and the release of ATP. So if you notice, what do you have to start respiration? You have to have energy to make energy, and that's the whole, you have to have money to make money. So in the process of glycolysis, so essentially we have 3 steps in respiration. The first step is glycolysis, and in the process of glycolysis, this is where you're going to take glucose, that 6-carbon sugar, and break it into 2 3-carbon pyruvates. And it makes a couple things. So it does make ATP, but primarily it makes NADH. Okay, and from that very first slide, the NADH is basically a hydrogen carrier. And if you notice, every single time it carries these hydrogens, what does it make? ATP. So really, what's making the most ATP? Things like NADH and FADH2. So these hydrogen carriers, these places that are being oxidized to carry this hydrogen, is actually what is causing more ATP production. So again, like I said, it's in 3 steps: glucose, and then pyruvate oxidation, and then the Krebs cycle. So notice that most of the ATP is being produced in the Krebs cycle. But what do you have to get past glycolysis? What do you have to be able to pull into your body enough of to where you can go through a full respiration instead of not going through a full respiration and ending up with muscle cramps? Oxygen. Okay, that's where this pyruvate oxidation, that's why you get muscle cramps. Okay, if you don't have enough oxygen, your body's going to go through glycolysis constantly. And that glycolysis will start making pyruvate. That pyruvate will go to a lactic acid fermentation. That lactic acid sits in your muscles, and it's like putting peanut butter on top of your fingers when you try to pull them back and forth, right? If I take your fingers and I keep them like this, and I say, okay, move your fingers back and forth, you can do them pretty easily, right? Okay, pretty easily. Okay, now I take some peanut butter and smear it all over your fingers, and smell on top and on bottom, okay, and then I tell you to go like this. It would be much, much harder, right? Possibly. It would be really much grosser. That's basically what lactic acid does to your muscles. Your muscles are relaxing and contracting, relaxing and contracting. Essentially what happens is that those fibers those muscles have get that lactic acid. That lactic acid is a sticky substance. And that's what causes that burn, that muscle cramp. Okay? Because you're running out of oxygen. And so because you're running out of oxygen, it can't go to the Krebs cycle to produce ATP. So it has to go to, it doesn't even go to oxidation. It goes pyruvate to fermentation. Okay, so here's the steps of glycolysis. I know it's really small. I do like the way the book does show it. It shows it, and it'll have this picture over and over again. So right now, glycolysis is where, so this is the mitochondria. Is glycolysis actually in the mitochondria? No, because look, right here, it says it's on the outside, right? So glycolysis actually happens in what's called the cytosol, or the liquid part of the cell, the kind of the gelatin part of the cell. No, you do not have to know all these steps of glycolysis. Okay? This is not Mr. Cox's class. I'm not going to make you learn all that. The AP board doesn't say you have to learn all that. Now, have you learned all this in Cox's class? Probably so. Or at least memorized it. I remember when I was in AP biology, this was something you had to memorize.
You gave us a text and we had to write down
every single step. I was actually just going back to my parents, like in my parents' house, and I've got this box of stuff I saved from high school, and I have a manila folder with this entire step drawn out. The AP board says now, the college board says now, this is not one of the required parts. Okay? You do need to know this idea that glucose is going in, that you have a step right down here of pyruvate, and that you have to use ATP, and that you also do create 4 ATP, but you use 2, so technically you get a net gain of 2, as well as you do create lots of NADH. Okay? So again, the key parts here is you do need to know this idea that ATP must be used, that you do use glucose to start it. It is on the outside. It is on the outside of the mitochondria. That this glucose, this 3-carbon glucose, does split into 2 3-carbon pyruvates. And that even though you use 2 ATP, you create 4, so you have a net gain of 2 ATP. Okay. And then finally, you produce NADH. And what's the purpose of the NADH? To carry the hydrogens to create ATP. Okay, so those are the main things. So again, you don't have to memorize these steps, but I do want you to understand what's going in and what's coming out. Okay? And why those are being created. Okay, because that's really what the test is going to ask you. The test is not going to ask you, go tell me on step number 4, what is being produced in glycolysis. It's not going to ask you that. It's going to say, in step number 4, NADH is being produced. Why? And so then you need it'll be a multiple choice question or a first response question, and you need to be able to answer the idea that that hydrogen that the NADH is carrying is going on to produce more ATP. So here is the process of glycolysis all broken down. This is actually two images. This is 2 images from 1 image in your book. The book does a really good job of truly showing how these different processes go. But again, like I said earlier, you don't have to know all the different steps. But I do want you to understand that it is being catabolic. What's catabolic mean? What is it doing to this glucose? It's breaking down that glucose. So it's catabolic, it's breaking down glucose. You do have to understand that it does occur in the cytosol, okay? And again, remember the cytosol is a gelatinous that's within the cell. You do understand that glucose does turn into pyruvate, and that you use 2 and you make 4, so you get a net gain of 2 ATP. And then also it does produce NADPH. I should put that on that slide, but I didn't. Okay?
But it produces
NADH, sorry. Okay. This is also where you create your water. Not nearly as important, but this is also where you would create your water. 2 of the waters. Okay. The next section, what? There's a lack of heat. Nice. The next section is the Krebs cycle. Again, you don't have to memorize the Krebs cycle, but you do have to know what's going in and what's coming out. Okay. Remember last time we talked about these cofactors, these coenzymes. One of the cofactors you have to use in the Krebs cycle is called coenzyme A. Okay. That coenzyme A is actually what starts the Krebs cycle. So again, we've got glucose. That glucose is being broken down to pyruvate. Okay, that glucose is being broken down to pyruvate. That pyruvate then is being carried through pyruvate oxidation, which is basically when it adds the acetyl-CoA to it, and it adds it into the Krebs cycle. Okay. Now, where are we in the cell? In the mitochondria. We actually have gone through 2 membranes of the mitochondria and gone into the inner membrane of the mitochondria. This inner membrane is within the folds of the mitochondria. Okay? So again, this acetyl group, these 2 acetyl groups are combined with the 4-carbon, which is called oxaloacetate. Again, not necessary for you to know that, but you do need to know a couple things. You do need to understand that there's a cycle here. Okay, per 1 glucose, how many times does the cycle go through? 2, because we're taking 1 glucose and breaking it into 2 pyruvates, and that pyruvate is going through. It's going to go through both pyruvates. And again, what must you have for this to even go into the Krebs cycle? Almost. Oxygen. Yeah, you have to have O2. Okay, you're going to produce ATP, and you're going to produce a good amount of ATP, ATP carriers in the Krebs cycle, but not a whole lot of ATP. So you do need to know that you have to have O2. Okay, so O2 has to go into this. And again, if you don't have O2, it's going to lactic acid fermentation or alcoholic fermentation. But you do need to know, again, look, we've got NADH here, we've got another NADH here, we've got another one, well, that's actually FADH2, which is the same thing. You've got another NADH here. Okay, every single one of those is going to carry a hydrogen. Every single hydrogen is going to create 2 ATP. So that's a lot of hydrogen being produced. So that's 1, 2, 3, 4. So you're going to eventually, from those four hydrogen carriers, you're going to create 2 ATP. So you're actually making 8. Well, it's not really 8 because you're actually going to go through the Krebs cycle twice. So you're actually making 16 ATP. So it's very efficient way to make ATP. But we haven't made enough yet.
So for 1 glucose, it's 16 ATP?
We're not done yet. We're actually going to end up making 32 ATP at the end.
So 1 glucose goes to 2 pyruvate goes to 16 ATP?
In the long run, yes. Yeah. We're actually going to end up making 32 ATP, but we have to get more hydrogens here in just a second. Okay. So again, we've gone from the cytosol with glycolysis to taking glucose and making pyruvate and a couple of NADHs. Okay. We're now taking that pyruvate, we've oxidized that pyruvate. We've gone into the mitochondria. We're now inside the very inner section of the mitochondria. The Krebs cycle is going to make 3 NADHs and 1 FADH2 and some ATP. Okay? And also notice it does have a byproduct of carbon dioxide. So again, this is where the carbon dioxide and the water are coming from. The water is coming from the glycolysis process, and carbon dioxide is coming from Krebs cycle.
What are the carriers?
NADH and FADH2. NAD and FAD. It's all about the NADs and the FADs. I don't know. So again, this is the Krebs cycle. So let's see if y'all can do this. Ready? If this, oops, if that right there is that 3-carbon pyruvate, what are we going to pull out here? Nope.
No, it's clock.
We're going to pull out. That's going to be actually, it's going to be NADH right here. Okay. So if, again, that's your Krebs, sorry, that's your pyruvate, NADH, sorry, blah. Yeah, NADH here. It's going to go into the Krebs cycle. This is the Krebs cycle. What is this little 2-part molecule right here? Carbon dioxide. What are these 3 right here being pulled out? NADHs. What is this 1 being pulled out as well? FADH2. And then what is this? ATP. Okay. So again, when oxygen is present, it does go in the mitochondria. It requires 2 turns for every 1 glucose, for every 1 step of glycolysis. And again, it does produce 2 CO2s, 1 ATP, 3 NADHs, and 1 FADH2s. Yeah. Well, it can be NADPH. It's just if it's phosphorylated or not. So it's either way. You ready for a song? Okay, we're going to introduce this with a song. It's awesome. This is Hazel's sister. Blowing my teeth out. Okay.
So I have a question. I have a quick answer. Okay, so NADH goes into complex 1? Yeah. And then NH bonds, right? Right. Okay. Sure. But what about this complex 3? It kind of confuses me. There's a lot going on in there. The electrons do split, they don't split. I don't know. There's like a million things to learn. First we learned glycolysis, then gluconeogenesis, then citric acid cycle, and now electron transport chain. Gosh, I just can't do this. Like, I'm just so tired. The finals are coming up. I just can't. Just stop. Please. Guys, let's calm down. Relax. Let's just close our eyes. Go to a tropical place. Pretend we have pina coladas, fruit juices. Hey, I think I hear music.
Transfer from the matrix to the I am space. Electron transport chain, you make ATP with your proton gradient and coupling with ATP synthase. Electron transport chain, you make ATP. Without you, I would die. Tonight, hey, complex 2, FADH2, turns into FAD and 2H pluses.
mics mean that they got professionally recorded.
Thank you, ladies.
Recording studio.
Wait, is that you?
They could have like added like.
Best thing ever, right? So again, those girls are carrying those electrons across the chain. So again, it's the ETC or the ETS, electron transport system, electron transport chain, whatever you want to call it. But this does all occur still in the mitochondria. Okay. So again, remember we were in the matrix, and the matrix is this middle part right here. Okay. The matrix is this middle part. This is where the Krebs cycle is. And the Krebs cycle is producing all these NADHs and all these FDH2s. Sure, it's making some ATP, but we haven't made a whole lot yet. Okay? But notice it is carrying a lot of H's. And every single one of these H's, remember at the very beginning we talked here, that every single of these H's is actually also carrying a what? Electron. So those little balls that are passing along, okay, those electrons will be passed through each of these complexes. Now notice, on their little video they have descriptions of each of these. But is it labeled here? No. So do you have to know each cytochrome C, cytochrome 1, whatever it is? No. But do a little kind of side note, a little an aside. One of the things that we know, one of the ways we can show that there is an evolutionary pattern within biology is that if you take the cytochrome C, okay, the cytochrome C that we have in our mitochondria is the same cytochrome C that a blue whale has in his mitochondria, the same cytochrome C that a plant has in its mitochondria, the same cytochrome C that a bacteria, okay, well not bacteria, but a protist would have in its mitochondria. Okay? And again, why not bacteria? What? They're prokaryotes. They have no membrane-bound organelles. So, and again, a mitochondria is more membrane-bound. So that's the same mitochondria, same cytochrome C. So what we can do is we can we can look at the protein structure of that cytochrome C. And that cytochrome C has a very specific this amino acid plus this amino acid plus this amino acid all the way through the primary structure. And we can look at the primary structure, and what we can do is we can compare the primary structure of cytochrome C for us and the primary structure for the cytochrome C for shrimp and find that that primary structure is not far off. Okay? So if you did a table, and we're going to look at this later on with cladograms, but if you do a table and you look at the primary structure of cytochrome C, the difference between us and, like, let's say chimps is about 2, maybe even 4. Okay? The difference between us and, I don't know, what, kangaroos or something is like 8. The difference between us and you go all the way down to plants, you go all the way down to a protist, it's going to be much larger. But there's a slow decline in comparison, and the number that are the same, the number of amino acids that are the same. So what that's showing us is as we become more and more advanced, as we become more and more of a multicellular, specialized creature, that our cytochrome C has changed, but it's basically been the same all the way through. So it's one of those evolutionary, not mechanics, but evolutionary ideas. Okay. Back to electron transport chain. Sorry, that was a side. Basically, the electron is being passed through. And every single electron is being passed. Every single time an electron is being passed through, what's being shot out? Hydrogens. Okay? So these hydrogens are being pushed out. So electron goes through, and basically it's this idea. Electron, remember, is negative. Hydrogen is positive. So basically, what it's doing is as it's shooting through this secondary structure right here, right there. Okay. As it's being shot through this first protein, it's negative, right? So it's actually going to pull these extra hydrogens through. Okay, it's kind of like you have this like really strong magnet. You've got let's say a north side magnet being pulled across. That south side magnet will want to come with that north side magnet if it's strong enough. Well, that electron is very, very strong, so it's going to pull this through. But before it actually, the hydrogen connect to that electron, it will pull it through that membrane and sit into what's called the inner membrane space. Okay, and it does that over and over and over again, to where what we have is that we have a very high gradient of hydrogens on the inner membrane space and a very low hydrogen gradient on the inside of the matrix. And we know if it has high and there's a low amount, what happens? Diffusion. Okay, so basically what it does, it diffuses from the high inner membrane space to the low matrix, and it starts turning what's called the ATP synthetase. You remember that on the video where the girls are spinning around? That's essentially what it is. It's a rotor, okay? And every single time a hydrogen pushes through that, it rotates that rotor, and it rotates that rotor, and it rotates that rotor. And every single time it does that, it's going to produce some ATP. So every single hydrogen that's being pumped here, it's going to go whoosh, whoosh, whoosh. And every single whoosh is going to create another ATP. Okay? So that's where the most is. Don't laugh at me for whoosh. That's what my AP biology teacher taught me, and that's what I'm going to teach y'all, because I remember the whoosh. All right? So every single hydrogen that's being pulled out is going to be pushed back in through that rotator, and every single rotation is going to produce a new ATP. Okay? So here's the kind of breakdown of it. It does incur what's called a cristae, which is the inner folding of the mitochondria. There is a series of oxidation reactions. Remember that oxidation reduction reactions, redox reactions, is the transfer of electrons. Okay? So every electron, just like that first picture talking about the steps, is reducing these electrons down to a lower level. And if you notice, on that previous slide, what's the final place that those electrons end up? What is the final electron acceptor? Can y'all see it up here? And there? You all see it on the front row? Oxygen. You do need to know that the final electron acceptor is oxygen on cellular respiration. So that final electron acceptor is oxygen on cellular respiration. It does then create water. So that water we will then breathe out. Our plants will breathe out or whatever. So final electron acceptor. Okay. The first donator is the NADH and the FADH2. The final acceptor is the O2. Do notice that it does occur, again, in the mitochondrial membrane, but it does slowly release, okay? Because you don't want to heat up too much. Remember, heat is useless energy. You can't really do a whole lot with heat, so you don't want to lose it due to heat. This is the step what's called chemiosmosis. This is this ATP synthetase, okay? And again, it is a rotor-type substance. Every single time we have a high concentration, it's going to go to the low concentration. And every single time a hydrogen whooshes through here, it's going to rotate this head. This is a catalytic head, meaning that this is a catalysis reaction. This is actually where ATP is being produced from ADP. So again, this is where we create new ATP. Okay. And again, the hydrogen will whoosh through. It'll rotate this rotor. Hydrogen goes all the way through, which then phosphorylates an ATP. This is it is coupled, meaning that as things are being used, it's being recreated, and it's being used, being recreated. ATP is being used, forms ADP, and it's being reused and forms ATP. Okay, it does produce between 34 to 36 ATP, and it's not very highly efficient though. That's the only issue, because it only converts about 38% of the energy we eat to ATP. Where's the other 62% going to? Heat, right? So that's the reason our body actually stays warm, is because we're only converting 38% of what we eat into usable energy. The other is being released as heat. It could also be used as storage or anything like that. Okay, so again, we're back into glycolysis. If glycolysis doesn't have oxygen to go onto the Krebs cycle, we go into fermentation. There's two main types you need to know: alcoholic fermentation and lactic acid fermentation. Alcoholic fermentation, it's used for wine making, for beer making, that kind of stuff. When you brew beer, you literally add yeast to it, and you basically pull the sugars out of the grains, and then that's a bunch of sugar. You put some yeast on the sugar. The yeast eats the sugar, and it produces alcohol. Okay. Whereas on yogurt, you get milk. You add bacteria to that milk. The bacteria start digesting that down. You pull all the oxygen out of it, and you start producing things like cheese, yogurt, and also chemicals like acetone and methanol. So again, this is actually one image that I've broken into two different images. These are the different steps of fermentation, both on the alcohol fermentation as well as the lactic acid fermentation. You do go through glycolysis. It does produce 2 ATP, and it does produce carbon dioxide if it's doing alcohol fermentation. If not, it then produces lactate. And again, this is in your muscle cells. This is also what bacteria do, those kind of things. Okay, so it doesn't produce a whole lot of ATP. There's not a whole lot of benefit. So again, when you're producing lactic acid, it's because you're not breathing enough. Okay, and it's either because you've worked so hard that your body cannot inhale as quickly as it needs to, or intake oxygen as quickly as it needs to use as much ATP as you're using. We talked feedback mechanisms last time. One I did not bring up to y'all is the idea of feedback mechanisms in your body too. So like things like insulin and glucose, those kind of things. But another one is pregnancy. Okay, the reason why pregnant women don't have cycles is because progesterone, and this is also the reason why contraceptives work the way that they do, is that progesterone is known as a pregnancy hormone. Okay, and a lot of contraceptives like produce. Well, you'll either take a pill that is progesterone based or estradiol based, and this progesterone, those estradiols, is a negative feedback. Basically, what it's doing is it's tricking their body into thinking they're pregnant. If they think they're pregnant, then they don't cycle. Okay, and if they think they're pregnant, they can't conceive again, and that's the whole contraceptive part of it all. So there are feedback mechanisms for this. Notice that if citrate is produced, it does inhibit. There's a lot of inhibitors, but also if you have a whole lot of ADP, okay, it's going to go start here, and it's actually going to start producing these ATP again. But if you have a whole lot of ATP, it's going to inhibit the part of the glycolysis. So there's more feedback mechanisms to either turn on or to turn off this process, because you don't want to start wasting glucose if you don't need to. If you have enough ATP, it's going to get through it. Okay. I want to kind of show you this. This is kind of a cool little video. Remember we talked about endosymbiosis and this whole membrane thing. This is a white blood cell, and there's a piece of bacteria in this. And just kind of watch it. It's kind of cool. The big object is the white blood cell. The little object is the bacteria, and it's literally chasing the bacteria. And I think the little things are actually red blood cells. Everything else floating around.
Bacteria like running away.
Yeah. And it's smart too, because it was like, oh, I'm gonna go there, and then it just ingested. So we call that again. This is that phagocytosis. This is pulling in large objects.
How's the white blood cell know where it's going?
Good question. I don't know. It's got some kind of stimuli, some kind of senses. So you could almost say that the white blood cell itself is within its own organism, because it's responding to stimuli, right? It can reproduce. It can go through mitosis. It doesn't really develop. That's the only problem. It's basically a white blood cell is a white blood cell is a white blood cell. There's different types of white blood cells, but it doesn't grow into different types. It just is either a T cell or a B cell or whatever. But other than the stimuli and the response to stimuli and metabolism and reproduction, it's almost a living organism, which is kind of cool, based on that video. So evolution in prokaryotes. Prokaryotes without mitochondria stop at glycolysis. So again, prokaryotes can only produce things like alcohol fermentation and lactic acid fermentation. That little video again is this idea of endosymbiosis. But some of the first prokaryotes, this is actually an image from your book, use sulfur as a final electron acceptor. Whereas what do we use? Remember? Oxygen. Okay? So we have to have oxygen to start that whole process, but that oxygen is then used later on to collect that last electron. So the initial prokaryotes can actually use sulfur as a final electron acceptor. So things like archaeobacteria can live in high sulfuric areas and can survive just fine.